Many of our bodily processes function in an oscillatory manner. The most obvious of these is our sleep cycle, a steady cadence with a time period of about 24 hours. But where does our body's time come from? The linchpin behind these physiological functions is the circadian rhythm, from circa meaning around and diem meaning day. The circadian rhythm is your so-called body clock, which cycles your brain through feeling sleepy and alert. The circadian rhythm isn't unique to humans. It can be found in other mammals, bacteria, and even plants. It's really important for life, and its interruption can cause adverse health effects including cancers, cardiovascular disease, and ultimately premature death. So how does it work? Well, the mammalian circadian rhythm is self-generated. That is to say, it exists even without environmental signals. Studies have shown that even after isolating a subject from light, they can still maintain consistent activity patterns. In fact, further work proved that the circadian rhythm has genetic origins. But first, a word about genetics. Genes represent sections of DNA which code for certain proteins. DNA itself is made up of two strands of nucleic acid chains, joined together with pairs of what are called bases. However, when DNA replicates, there can be errors in the arrangement of these bases. This is called a mutation. A study in 1994 found one such mutation in mice. A mouse with a mutation had a 25 circadian period, and so did all of its descendants. But how do you find where the mutation occurred? Well, you can try overwriting sections of the mutated genes with healthy DNA. These sections are called transgenic gene fragments. If an injected fragment caused the mouse to revert to a normal rhythm, you know that the broken section must be within that length of DNA. When they found the perfect gene fragment through trial and improvement, the scientists had isolated the gene for controlling the circadian rhythm. They named it the clock gene. The actual mechanism of the process is as follows. The clock gene codes for, you guessed it, the clock protein, and together with another protein, BMR1, forms a heterodimer, hetero meaning different, di meaning two, and mer indicating that it's a polymer. The clock BMR heterodimer activates even more genes to construct proteins in your cells by binding to a specific section of DNA called the E-box, or enhancer box. Here's where it gets interesting. Clock BMR activates the production of two types of protein, per for period and cry for cryptochrome. Per and cry accumulate in the cytoplasm around morning, but eventually they move back into the nucleus and cause havoc as they actually inhibit clock BMR function. Per cry can form a heterodimer of their own which can displace clock BMR from the enhancer box, preventing protein production. But wait, remember how clock BMR activates production of per cry? As the concentration of per and cry increases, its own production is slowed, creating a negative feedback loop. Then, throughout the night, per cry eventually decays and allows clock BMR to be synthesized again. This produces per cry again and the cycle is repeated. This entire cycle takes about 24 hours and therefore makes up your primary body clock. So what do these molecules do when they're active? Clock BMR affects the production of serotonin and 10 other neurotransmitters, chemical messengers used as signals between neurons around the body. These chemicals are what cause you to feel tired and drowsy. Not only that, clock BMR also activates 15% of all protein synthesis, including important proteins that affect insulin production and other metabolic processes. Disrupting the rhythm can therefore increase risk of diabetes and other conditions. We hope that you now have a better understanding of the biological clock inside your body and how complex and important it is for your survival. Thank you for watching.